In randomized controlled trials, we randomly assign participants based on exposure X to a control or treatment group, and then we assess their different response Y. This artificial randomness allows unbiased comparisons to test if X influences Y, and this is precisely possible because we can control the variable X. But what if we cannot? In such cases, we turn to instrumental variable methods, and one powerful approach is two-sample Mendelian randomization. With two-sample Mendelian randomization, we bifurcate our study into two distinct samples, one for the exposure variable and another for the response. Through conducting genome-wide association studies for each, we associate single nucleotide polymorphism, SNPs, with both phenotypes X and Y. Then, based on Mendel's second law of heritance, we know that these SNPs are naturally randomized, so we can use them as our controlled instrumental variable. And here's the magic. If the SNPs that are strongly associated with X show also correlative associations with Y, then this is an indicator that X and Y might indeed be interconnected. But hold on. To make this work, we are making some deep assumptions about our SNPs, or instruments, and its relations with the other variables. The first instrumental variable assumption assumes relevance. That means our chosen SNP strongly associates with X. The second instrumental variable assumption assumes independence. No co-founders meddle with the SNP outcome association. And the third one, the exclusion restriction assumption, states that the SNP only influences the outcome through the risk factor of interest, X. And why are these principles crucial for causal inference? Let's visualize how our Gibus betas are generated using this causal diagram as a reference. We can assume the betas follow a normal distribution, with a mean determined by the sum of the paths connecting the instrument with the X and Y variables, respectively. Since there is only one path connecting them under the instrumental variable assumptions, we can estimate the exposure-response effect using the Wald ratio of our observed betas. Additionally, when assuming no measurement error, it's a very standard practice to expect the variance of the instrument exposure effect to be approximately zero. Hence, we compute the variance of our exposure-response estimate as a ratio between the variance of the instrument-response relationship and the effect of the instrument on exposure. Now that we have laid down the base assumptions, let's explore the statistical methods used to determine the XY effect in Mendelian randomization. In a simple scenario with, say, 4 instrumental variables or SNPs, one might initially think of using a basic linear regression. In other words, plot the effect of the instrument on Y against its effect on X, and voila! The slope of the regression line tells us if the relationship is positive, negative, or null. However, I'm going to tell you a secret. No one ever uses a basic linear regression. Why? Simply because not all instrumental variables are equally precise. To account for this, the inverse variance weight is a method that assigns weights based on the inverse variance of the estimates, ensuring that more precise instruments carry more influence in the estimation of the slope. And what if some structural assumptions are broken? Let's say the third instrumental variable assumption, which assumes that Z and Y aren't directly related through paths over the X. This would be called horizontal pleiotropy, or simply pleiotropy. If pleiotropy is random and unrelated for each instrumental variable, it might not be too troublesome, as the inverse variance weight commands relatively well-balanced pleiotropy. However, when our SNPs are all entangled in similar pathways, which is very common in biology, by the way, that may produce an unbalanced pleiotropy where many SNPs are wrongly pointing to the same direction. The easy solution, Egger regression, a statistical method that allows adjusting the intercept, addressing the problem head-on. But as you might imagine, Egger regression isn't a cure for all. When measurement errors creep in, violating the norm assumption, detection power may wane. Random errors in X can introduce what is known as a regression dilution bias, flattering the curve and making it harder to detect a true causal relationship. Linear assumptions can also be a hurdle. Both inverse variance weight and Egger regression assume a linear relationship between exposure and response, but this is not necessarily always true. Another big problem for these methods is the violation of the inside assumption, which happens when there is a relationship in the strength of the instrument's effect on X with the instrument's effect on Y that doesn't go through X. In this case, changing the intercept is not sufficient, as the slope of the curve is also affected. Even worse, for the Egger regression, the simple breakdown of this assumption, even for one point, can entirely alter the method's result. What do we do for those two cases? A popular non-parametric alternative is the weighted median. It considers the precision of each instrument and makes inferences based on the median weighted SNP. While it sacrifices some power, it's less sensitive to assumption violations, requiring only 50% of the weight to come from viable instruments. But in summary, embarking on a two-sample Mendelian randomization journey is no small feat, and you have to be careful in designing a strategy. First of all, define a relevant problem that includes an exposure with certain genetic basis, considering measures as heritability and proposing hypotheses where is the cause of an effect variable. Then, define proxies or measures for each, critically evaluating their appropriateness to the analysis. Afterwards, find two US studies using them. Make sure they are or can be stratified or blocked for potential measure confounders, such as the subpopulation genetics. Select those with the largest sample size to reduce potential weak instrument biases or findings by chance, and ensure that the cohorts are exchangeable, coming from the same compliers, but independent from each other in terms of cohorts. So, different potential biases can go towards the null. The next step is probably the most critical one, defining the genetic instruments of your analysis. I personally recommend that you obtain your instruments from independent sources to the ones you use in your analysis, either by using different GWAS, biological databases, or your personal expertise. The priority is to select variants that are specific for X, even better and more informative if they are causally related. It is also interesting to make sure those variants don't impact survival outcome before the age of the GWAS is performed, so no selection bias is done. Once you have them, if you plan to perform the methods I mentioned, only select a subset of SNPs that are LD independent, so your regression is made by independent points. And finally, always perform multiple methods. Employ a variety of analyses that consistently validate your results. I also recommend to perform sensitivity tests and plots to determine which statistical method is the more appropriate for your case. Different figures, such as funnel plots, can help you determine if your SNPs present unbalanced pleiotropy or not. There are also statistical tests to assess this, as the Eggers-intercept test. Another interesting thing is to study heterogeneity, which can be observed directly in your scatter plots or by using a Cook-Cochran test. The important thing is that heterogeneity is going to tell you if other structural assumptions or pleiotropy assumptions could be broken. Anyway, and despite of your final result pointing out causality or not, always remember that strong conclusions should only be drawn when multiple sources of evidence align. Causal inference in the realm of observational data demands caution, and triangulation with other experimental methods remain imperative. Let me know in the comments if you have additional questions or points to address. But anyway, thank you for watching.